My name is Gorka Lasso. I am a research assistant professor at the Department of Microbiology and Immunology in Albert Einstein College of Medicine in New York, and today I will walk you through our poster. Ebola viruses belong to the filoviridae family. These viruses are known to cause recurrent outbreaks of severe disease in humans that include mortality. Until recently, 5 Ebola virus species were known, being Zaire Ebola virus the prototypic Ebola virus species. Moving forward, when referring to Zaire Ebola virus, I will also use its acronym EBOV. With the exception of Residue virus, all other Ebola viruses have been associated with severe disease in humans. In 2018, a new Ebola virus species named Bombali Ebola virus was found roosting inside the houses of locals in Sierra Leone. This shows that humans can be exposed to these viruses and the necessity to study its pathogenicity. However, it is not clear whether Bombali Ebola virus is pathogenic in humans, since there have not been any reported case of human infection. Viral entry is an essential step for viral infectivity, and the Niemann-Pick C1 protein, or NPC1, is a cholesterol transporter that is known to act as an essential receptor for Ebola viruses and other viruses within this family. In vitro analysis has shown that Bombali glycoprotein interacts with human NPC1, driving viral entry into human cells. Here, our goal is to further characterize Bombali entry and assess the potential risk of human infection. For this, we experimentally study viral entry using a recombinant vesicular stomatitis virus, or VSV, bearing the Ebola virus glycoprotein on the surface. In this case, we use 2 different recombinant Ebola viruses. One bearing the Zaire Ebola virus glycoprotein, shown in blue in the central and right panels, and a second one bearing the Bombali Ebola virus glycoprotein, shown in orange. The middle panel shows a binding ELISA where we study the interaction between the viral glycoproteins and human NPC1. The right shifted orange curve of the VSV with Bombali glycoprotein shows the lower affinity of this glycoprotein with respect to Ebola for human NPC1. The right panel describes the infectivity assays using human cells. These results are in accordance with the binding data, since we observe a lower infectivity for Bombali than for EVO, and such differences are no longer observable when overexpressing human NPC1, which shows that the differences in infectivity are due to the differences in binding affinities observed by ELISA. We use the X-ray structure of the interaction between Zaire Ebola virus glycoprotein bound to human NPC1 to infer interfacial residues in the Bombali glycoprotein whose identity differ. We identify 4 residues at the interface located at positions 146 and positions 148 to 150. Following this, we mutated each of these positions in the X-ray structure to assess the energetic contributions of each of these mutations upon binding complex formation. For this, we employ 3 distinct and complementary tools: FoldX, FlexDTG, and free energy perturbation, also known as FEP. All 3 methods consensually predicted proline to serine mutation at position 146 as the most destabilizing amino acid change at the interface. We experimentally validated that proline for serine at position 146 drives the difference observed in binding ELISA and infectivity assays. EBOV with serine at position 146 shown in light blue, behaves like Bombali ebolavirus in orange, and Bombali ebolavirus with a proline at position 146 in green behaves like Zaire ebolavirus. We further analyzed the molecular dynamics trajectory of FEP for Zaire ebolavirus glycoprotein and the mutant bound to human MPC1. The backbone nitrogen of serine at position 146 acts as a hydrogen bond donor, forming an interaction with the backbone oxygen of the human MPC1 aspartate at position 501. This hydrogen bond brings closer the respective protein backbones and causes a kink in the human MPC1 loop that mediates key interactions with Zaire ebolavirus glycoprotein. Altogether, we have observed that the reduced Bombali glycoprotein interaction with human MPC1 attenuates entry into human cells. Structure-based computational analysis predict proline for serine at position 146 as destabilizing upon complex formation, and this has been validated using both binding and cell-based infectivity assays. Finally, FEP trajectory analysis suggests that viral glycoprotein proline to serine at position 146 induces a conformational change on the human MPC1 protruding loop 2, which might affect the binding of the mode of binding.